Hello, my friends. In this video, we'll be taking a look at the DNA results, G25 results, haplogroups, and the health and traits of an early Ashkenazi Jew from Erfurt in Germany. This is the location right here. You might not know this little bit of lore, but the reason they are called Ashkenazi Jews is because one of the descendants of Japhet was called Ashkenaz, and he lived in Europe. So these Jews are living in the lands of the descendant of Japheth, Ashkenaz, and that is why they are called Ashkenazi Jews, because they're European Jews. So these are early European Jews from Germany. I'm going to make probably a couple more videos on these Jews. Make sure you watch the previous video on Ashkenazi Jew from Germany, from Erfurt. This is the second sample. I am, by the way, in case you are wondering, I am purposely picking out these samples who are light pigmented because I have a little bit of it's not that I have an agenda, it's that I have a interest in Jews who are light in color. It's just very interesting to me. So we're going to look at another sample right now. This individual is also quite light in terms of pigmentation. He has I think it's a female. Yeah, she's got green eyes, and her mitochondrial lineage is K1A1B1A. Very, very interesting mitochondrial lineage. First, we're going to start with her results with G25. So, with G25, if we include the Jewish populations, she's closest to Ashkenazim from Poland, followed by Maltese, followed by Ashkenazim from Germany, Lithuania, Ukraine, Belarusia, Russia, Romania. Then Italian Jews, Italians from Calabria, Sephardic Jews, Sicilians, West Sicilians, Greeks from whatever that is, East Sicilians, Greeks from Crete. So she's quite similar to various East Mediterranean populations and Ashkenazi Jews. She's very much an Ashkenazi Jewish, very similar to various Ashkenazi Jews. But I decided to try to model her without Ashkenazi Jews um, in the reference populations. So let's try to see a model without any Ashkenazi Jews or Jews in general, European Jews in general, in the reference groups, in the reference populations. So if we remove those from the reference populations, we see the closest populations is Maltese. Um, Maltese, they actually speak a dialect of Arabic, so this is actually an Afro-Semitic group. That's a Semitic group that she's closest to, which speaks volumes about the ancestry of Ashkenazi Jews, because some people say they're Khazars, some people say they are European converts, and obviously this is not the case when the closest population is Maltese. And yeah, I mean, it's very obvious when you look at the results, the genetic results of these samples from Erfurt. It is very clear that they are nothing like Germans in terms of autosomal ancestry. Let's look at the single mole population oracle, and with the single mole population oracle, she is getting modeled as a mixture of 60% Albanian, 27% Samaritan, so that would be Middle Eastern or Israelite, and 12.4% Tunisian Berber. I'm not sure where the North African admixture comes from. Maybe it is also part of the Israelite ancestry. So I think it is. I think it is part of the Israelite ancestry. I think you have to add the Samaritan plus the Tunisian together, and that together would be Israelite ancestry. So I think in total she has 40% Israelite ancestry and 60% Southern European ancestry. That's how I would view it. And she does not have any Northern European blood. Let's see if we reduce it to 5 populations. Is she gonna score anything from Northern Europe? No, she does not score anything from Northern Europe. She scores 47% Albanian, 29% Samaritan, 10% Bosnian, then some Berber and Tunisian. So she does not score anything from Northern Europe whatsoever. What if we remove the reducing populations? Okay, here she does score a little bit of Northern Europe. She scores 0.8% Lithuanian, but yeah, she doesn't really have Northern European ancestry whatsoever, which is very, very interesting because she has very light pigmentation for traits. Let's go ahead and look at her traits. So first, we're going to look at Nashakot results, Nashakot calculator results, and we're going to start with that. So first, for eye color, she has green eyes. That's the predicted eye color for her at 35.6% likelihood. The second largest likelihood is blue eyes, and the third largest likelihood is blue eyes with an amber center. Hazel eyes, there's a pretty large percentage likelihood for hazel eyes as well at 15.9%, but most likely she has green eyes. And that's a very light prediction for somebody from this area. And she probably does not have brown eyes or darkest brown eyes. That is pretty much out of the picture. For hair color likelihood distribution, she's actually predicted to have dark blonde hair. Once again, this is very surprising for somebody who is so Mediterranean in ancestry to have dark blonde hair. The second largest group that she scores is light brown hair at 33% likelihood. And there is some chance of light blonde hair actually as well, and dark brown hair as well at 6%. But most likely she has dark blonde hair. Very, very interesting. For skin color, she is scoring white skin at 72% likelihood. Very, very interesting. And for hair texture, she is scoring actually kinky or curly hair. Wow, that is surprising. That is quite surprising. But it's quite all over the place for the hair texture. So it seems that the file maybe wasn't high quality enough to get a more accurate prediction. But she is scoring kinky or curly hair for hair texture. For nose shape, she is scoring rather Greek than snub-shaped nose, so more of a aquiline, long, and narrow nose shape, which is typical for Ashkenazi Jews. It is much the stereotype of what they look like. They have, they're very much stereotyped with having a Greek aquiline nose shape. Now let's look at the coloring related variants. So we see two light color variants here in Herc 2. From this, we already know that with snipper 3, she's going to score blue. She's got two light color variants here. Well, actually, we don't know that she's going to score blue, but she most likely is going to score blue. She's got blue haplotype 2. Yes, blue haplotype 1 as well. Yes, from this, already probably going to score blue. She's also got heterozygous genotype for blue haplotype 3. Yeah, probably going to score blue with snipper 3. For eye color, probably going to score blue. With Y-sec, she's going to score blue as well because of the two light color variants in this variation of BH2. Because for Y-sec, this is pretty much the only variation that matters. They don't really go polygenic like My2 or Snipper 3. No genotype for this variation of SLC24A5, but we can assume she's got two light color variants here. She's got two light color variants in both the relevant variations of SLC2445A2. Really good to see. Okay. And no ginger variants in MC1R, so no predisposition to be ginger. Let's check what she scores with Snipper 3. I am expecting it to be blue for eye color with Snipper 3. There is no reason for it to be anything other than blue, but let's check. Yeah, it's blue. So she's scoring blue eyes with Snipper 3. Alright. Let's look at the phenotype oracle. And the closest phenotypes to her is this, which is, by the way, in case you are wondering what's the difference between this phenotype and this phenotype on the second phenotype on the bottom, it's that the first has curly hair. That's the difference. There's a subtle difference here. So because hair texture actually also plays a part in the phenotypes you score. And she was scoring very high for kinky hair. So because of that, she's scoring this phenotype that has curly hair as the closest to her. And the third closest phenotype to her is this, which is also, I'd say it's also quite Jewish looking. I mean, it's very light and it's very blonde, but I think it also looks pretty Jewish. I'd say it's also a very Jewish looking phenotype. For the phenotype oracle, she's getting modeled as a mixture of 50% this plus 50% that. This on the bottom is curly hair. Then 50% this plus 50% that. This on the bottom has, I think it has straight hair. But this phenotype on the top has sort of curly-ish hair. Then 50% this plus 50% that. This is curly hair. This is, I think, straight hair. Then 50% this plus 50% that. Very, very interesting models. So all of them are kind of saying the same thing. She most likely looks like something resembling these phenotypes. Something between this phenotype and this phenotype, essentially. Alright. Should I look at the ethnic calculator results? Let's look at it. Let's see what she scores here. I will probably... Let me do this. I will zoom in. And I will pause for 1, 2, 3, 4, 5 seconds. Let you guys take a screenshot or let you guys pause the video. and look at it. Maybe just literally click pause the video, and I am going to move on because I don't really want to dwell on this too much. Let's go and look at the biomarkers results. We're gonna look at what she scores for the biomarkers. Let's reduce the reset button. Okay, so above average levels of vitamin D, really good to see. Above average levels of LDL cholesterol, which is really bad to see, really unfortunate. Below average level of HDL cholesterol, which is also really unfortunate to see. Below average level of glucose, which is good. Above average level of hemoglobin, which is okay. Above average blood pressure, but it's kind of okay because it's still in general average range. Slightly below average level of iron in the blood, so no hemochromatosis. Slightly below average sex hormone binding globulin in blood, so that's pretty typical. Slightly below average level of red blood cell count in blood. Nothing was found for number of base pairs or telomere length. Alright, so it looks like there wasn't that much data in the file for telomere length. And she's predisposed to be much shorter than average. So 1.8 centimeters shorter than average in height. Very interesting. So if the average white female is 165 centimeters, then she is predisposed to be 163.2 centimeters in height. Definitely quite interesting. Let's close that. Now let's look at the polygenic risk scores, and we're going to look at stuff that is essentially distant from 1, stuff that is not average. Everything that's average we are going to ignore. So she's got below average odds for arrhythmia and nodosum, really good to see. Below average odds for hemoglobin E disease. Below average odds for migraine. Above average odds for lupus. Below average odds for gout. Below average odds for thyroid cancer. Below average odds for eczema. Above average odds for cataracts. Below average odds for AMD. She's got above average odds for epilepsy. She's got above average odds for asthma. She's got above average odds for leukemia. She's got below average odds for vitiligo. She's got above average odds for myopia. She's got above average odds for corneal astigmatism. She's got above average odds for primary biliary cirrhosis. She's got above average odds for male pattern hair loss, which is not surprising because Jewish people are stereotyped as losing hair very early on. It's a stereotype that Jewish people lose hair pretty early. She's got below average odds for atrial fibrillation, below average odds for deep vein thrombosis, below average odds for cardiovascular issues. Really good to see. That takes into account both the homocysteine and cardiovascular panels. So that's really good to see. Below average odds for ADHD, straight up average odds for unipolar depression, high odds for bipolar type 1, and high odds for schizophrenia. Very interesting. So the things that she scored high for is bipolar type 1 and schizophrenia. That's quite interesting. And there is, I have heard that it is more common among Jews to have these types of mental health issues like schizophrenia and bipolar type 1. I'm not sure how true that is. I'm not sure. She's got higher odds of type 2 and type 1 diabetes. Alright. She's got lower than average odds of Alzheimer's and lower than average odds of multiple sclerosis. 2 risk variants for breast cancer out of 6. Typical. None of them, they're both in the BRCA, I think, but that's okay. 5 risk variants for testicular cancer out of 18, and she's got 2 of them are in the important variants in Ketal G. So she's quite cooked when it comes to risk variants in the Ketal G. But it's not that big of a deal because most West Eurasians have the same type of genotype here. So it's not that big of a deal, to be honest. You watching this video, if you are a West Eurasian, probably have the same genotype here. And you probably will not develop testicular cancer. So it's not that big of a deal. For celiac disease, she's got 3 risk variants for celiac disease out of 8. They're here. For GSS, no risk variants. For Crohn's disease, 7 risk variants out of 18. Alright. None of them are marked as important, so we can ignore that. For Raffenstein's, nothing found. For Parkinson's, nothing, no risk variants for that. Gilbert syndrome, nothing found. Basal cell carcinoma, nothing else. Looks like nothing was found for any risk variants for her. Really good to see, but we do have to watch out for, what do we have to watch out for? Bipolar type 1 and schizophrenia. And also type 2 and type 1 diabetes. So let's go through her results and see what she scores. Looks like for warrior versus warrior, she's definitely scoring more of a warrior phenotype, quicker dopamine reuptake and lower dopamine levels, higher stress resiliency. Um, just intuitively, it is not a phenotype that I would associate with schizophrenia because it doesn't make much sense. But I have seen quite a lot of research that actually does link this phenotype with higher odds of multiple mental health issues like schizophrenia. So I don't really know what to say. In my trait predictor, these variations do not play a role in determining the risk score for schizophrenia. So it doesn't play a role in the polygenic risk scores. But intuitively, it doesn't make much sense for me that the warrior phenotype would be correlated with risks of schizophrenia. But it does. Apparently, it does. According to some research, it does, which is very interesting. So essentially, she's more of a warrior. She's got quicker dopamine reuptake, lower dopamine levels, and higher stress resiliency, more activity of the COMT and MAOA enzymes. For DRD2, she's got higher activity, higher number of dopamine D2 receptor sites in the brain, and this is very, very robustly linked by research to higher odds of schizophrenia and bipolar, and that's why this is actually playing a role for the polygenic risk score in my trait predictor. So all of these DRD2 variations from this to this, they are all playing a role in the polygenic risk score prediction for the odds of schizophrenia and bipolar type 1, both these conditions. So her scoring higher number of dopamine D2 receptor sites in the brain, that makes sense. That is also linked to her scoring such a high score for both of these conditions. And she's scoring lower levels of 5-HT and serotonin, so she's got some predisposition to conditions such as anxiety and depression. Very, very interesting. All right, so for autism, she's got higher odds of autism. For lactose persistence, looks like she's got intermediate or higher odds of lactose intolerance, and she does not have any variants for European lactose persistence. Nope, doesn't carry any variants for European lactose persistence. So she's scoring. This is the score you will get if you got these three variations in your file, and you don't have any variants for European lactose persistence. This is the score you will get, essentially. She's got a predisposition to lower level of empathy in OXTR based on these four variations. Very interesting. So quite unempathetic. Definitely very interesting as well. Does she have any variants associated with higher level of empathy? Let's look. She's got one genotype associated with higher oxytocin and empathy. So there is one genotype that is associated with higher level of empathy in her genome, but there is also this variation which is associated with sociopath variants. This variation which is associated with lower level of empathy, and this variation to sociopath variants once again. So really, there's more variations that are linked to lower level of empathy than there are linked to higher level of empathy, and that's why she's scoring the way she is. So overall, her predisposition is to a lower level of empathy based on these four S&Ps. It's 1 versus 3. Okay. I'm not even gonna go through this panel because it's not very informative. I should probably make it more informative. Okay. For multiple sclerosis, she's got no risk variants for MS and HLA gene. Really good to see. Cardiovascular disease panel, she's got a predisposition to lower or intermediate odds of cardiovascular issues. We already could expect that because we've seen what she scores for the I forgot what it's called. Let me find it right now and I'll remember it. Cardiovascular issues panel, yes. So we've seen what she scores for the cardiovascular issues panel is below average odds for cardiovascular issues. So because of this result, we already know that she would be scoring quite good for the cardiovascular disease panel because that result is based on this. For the facial morphology panel, she's got 2 European variants and all the relevant variations in EDAR. Really good to see. So she's quite West Eurasian or European in facial morphology. She does have one HLA variant, HLA risk variant in the vitiligo panel, but we are probably going to see really good results for the HLA gene panel. I'm expecting really good results. She's got lower odds of missing teeth. She's got smaller nose size, higher odds of protruding nasal bridge, shorter mid-face length. Very interesting. No micropenis as well. really good to see. Better performing muscles, likely sprinter rather than endurance athlete, really good to see. And mm, no variance for pain hypersensitivity out of 2 total, really good to see. No risk variance for familiar Mediterranean fever. Unfortunately, there isn't much variance found in the file. It's not a very high quality file. Usually, you will find like my Trait Predictor will find like I think 15 or 20, but in this case, only 2 were found. But no risk variance here. And for the MTHFR panel, normal homocysteine levels, good genotypes here. Two good genotypes. So probably we are going to see good results for the homocysteine panel as well. Of course, we also have to look at the CBS gene panel to really know for sure, but we are probably going to see good results for the homocysteine panel as well. And rare diseases and traits panel, let's look. 2 risk variants or panic disorder, which is very rare. This is probably not a miscall because it's a homozygous genotype, so there is some predisposition to, looks like panic disorder. I found this variation on GWAS Central, and it has a pretty strong, I don't know how to say this. Um, it has a very high odds ratio, and it also has a very low p-value. So it's one of those variations that's very strongly linked to panic disorder. So in this case, that's quite a significant find. Very interesting that this sample has this type of a genotype. Unless it's a miscall, in which case, but I don't think it's a miscall because it's a homozygous genotype. I don't think it's a miscall. She's a carrier of risk allele for neural tube defects in spina bifida. 20 times increased risk of ankylosing spondylitis, but that is very common. I know it sounds very scary when you read 20 times increased risk, but it's actually very common. This type of genotype is like 5 or 6% of genomes. Like 5 or 6% of people score that, so it's not very unusual whatsoever. 5.2 times risk of rheumatoid arthritis. Okay, that's kind of uncommon, yes. And 2 risk variants for leprosy. All right, but I'm not seeing anything else here that's unusual. Single HLA-DQ8 haplotype, higher than average risk of celiac disease. So there are some variants in the HLA gene that this individual has that are predisposing her to risk for various autoimmune conditions such as the celiac disease. Okay, she does have 2 protective variants in HLA-C from HIV, which is really good. That leads to 90% reduction in HIV viral load. Really good. No risk variants for Canavan syndrome. Nothing was found for Crohn's disease, but this is really good. So she's quite protected from HIV, which is really good. Now let's look at the HLA gene panel, and here we see she's actually predisposed to intermediate odds of autoimmune disease. Wow, this is kind of concerning, and this might explain the very high score for type 1 diabetes. Actually, this might explain that, because the HLA gene panel does influence the polygenic risk score for type 1 diabetes. Let's look at the HLA genotypes and explore them a little bit in detail. Let's see. This is a good genotype. This is a good genotype. This is a good genotype. So HLA-A is looking very good. HLA-B is also looking really good. HLA-C, these 2 variations are not looking good, but HLA-C, this variation is looking pretty good right here. Now HLA-D, which includes DQ, DR, and DP, I'm not sure. This is a bad variation. Even though it says leads to a slight decrease in risk of type 1 diabetes, it is really quite bad because it's slight decrease relative to the like the ideal. Well, I should. How do I phrase this? How do I phrase this? I don't know. Brain freeze. It leads to a slight decrease relative to the worst case scenario risk of type 1 diabetes, yes, but relative to the ideal genotype, which is the most common genotype worldwide, it's actually a very much of an increase. And there's also this, which is really uncommon. So yeah, this probably was the biggest contribution to the intermediate or slightly higher odds of autoimmune disease. There was also this, which also contributed as well. There's this which contributed as well, but really this as well, high odds of lupus, this right here. contributed too. In HLA-C, these two genotypes contributed as well, but really, there's the biggest contribution, in my opinion, was probably this HLA-DQ8. I don't remember how much of a contribution exactly these variations play, but I think this variation was the biggest contributing factor to her scoring what she is for this panel, because her score isn't really good at all. Scoring intermediate or slightly higher odds of autoimmune disease as the largest percentage for the HLA gene panel is it's kind of like a warning. It's kind of a risk. All right, so for the MTRR gene panel, she does not have any risk variant. She has one risk variant here in this variation, one risk variant for spina bifida and disorders of intracellular colon metabolism. So that's 1 out of 6, very typical. And for CBS gene panel, wow, really good results. So she's scoring lower homocysteine panel. That's kind of what I was expecting, but yes, and it makes sense because she's got these genotypes lead to decreased homocysteine levels. She does have this genotype which is to higher and this genotype leads to. So I guess for the CBS panel, it's kind of like all over. Some genotypes lead to lower, some genotypes lead to higher levels of homocysteine. But overall, if we look at the results with the MTHFR panel, the MTHFR panel plays a bigger contribution actually than the CBS panel, which is kind of ironic because the CBS panel is like right here. It's located right next to the homocysteine panel, whereas MTHFR panel, I have to scroll all the way back to like wherever. I don't even know where. Where is it? I can't even find it now. Yes, but MTHFR panel actually plays a bigger role in this homocysteine panel than CBS panel does. Okay, so for muscular dystrophy myopathies, she's got, and essentially that's really good because that also contributes to the low score for various cardiovascular issues. That's really good. Lower homocysteine levels is good for your cardiovascular health. For muscular dystrophy myopathies, no risk variance for anything here, no risk variance for ADL, really good. For color blindness panel, no risk variance, NOP and 1SW, really good to see. For FTO gene panel, she's got predisposition to intermediate odds of obesity. It's really straight down in the middle. 25% for higher, 25% for lower, 50% for intermediate. That's really, really neat looking. And that's based on 1, 2, 3, 4, 5, 6 SNPs. So based on these 6 SNPs, she has a predisposition to straight up intermediate odds of obesity. Really cool to see. So looks like she's homozygous for fat gene variants here, heterozygous in these variations in FTO, but she's got these 3 variations where she's got lower BMI. So if you take all that into account, she's got straight up intermediate odds of obesity. For syncope panel, she's got based on 2 SNPs below average odds for syncope. In case you don't know what syncope is when you faint, you lose conscience based on loss of blood flow to your brain. It is usually psychological. Usually you encounter some psychological event that leads to, for whatever reason, your body is storing blood in your legs and it doesn't go to your brain, so you lose consciousness. I'm not sure why that happens to some people. She's got 2 copies of the farmer CLT-CL1 gene variant, which leads to a selective advantage in processing carbohydrate-rich diets. I don't know what is the ethnic distribution. I never bothered to look it up, of the ethnic distribution of the CLT-CL1 variations, farmer versus hunter-gatherer variations. Maybe I should do a video on this. You know what? I think I will do a video on this topic. I think I should. Maybe it would be a good video. But yeah, it would probably be a good video. But it's very interesting because Jewish people, they descend from Anatolian Neolithic farmers, from Natufians. They descend from all these populations that transitioned to farming really early on. So it would make sense for them to have these genotypes that help them process carbohydrate-rich diets. So I don't really know what is the ethnic distribution of this variation, but I could look it up and I could make a video on this topic. I just got a brilliant idea, I think. Maybe that's going to be the video idea for tomorrow. I don't know. I don't really know. Let's see. And for the blood group panel, she's scoring blood type A. Very, very interesting blood type. Most common blood type is blood type O, but we have indeed here found a blood type A. Although there's quite a big chance for blood type O at 10.8%, blood type B at 11.018%. Big thanks to you all for watching my video until the end. Make sure you check out the description of this video. There you'll find the link to buy the raw DNA file of the sample I analyzed here, buy my Trait Predictor. Very soon I'm releasing the update. It is going to be the upcoming days. With the update, it's going to be a sale. I think I'm going to make the price $20. The sale is going to last for probably a week. I'm not sure how long I'm going to make it last. Maybe longer, depending on how well this selling is going to go. Maybe forever. I don't know. But make sure you check out the description of the video. And thanks for watching. Leave a like and subscribe if you enjoy my content. Goodbye.